replicate without a mistake, no mutations. But some of them have one or two. But this guy makes a lot of them, okay? So here's the deal. Retrovirus genomes mutate very, very rapidly. Reverse transcriptases are prone to make errors, much so than polymerase. They bind nucleotides, they put wrong nucleotides into the DNA, they make mistakes, right? And they don't have proofreading. They don't have that 3' to 5' exonuclease activity. So whenever they put in the wrong nucleotide, that's a mutation. And they put them in a lot. RBTs incorporate the wrong base every 1,000 bases, not every 10,000, like eukaryotic cells. 10 to the 5th is 100,000. But every 1,000 bases, they incorporate the wrong nucleotide, causes a mutation, and that mutation has a chance of changing a protein on the surface or something that changes their antigenicity. So when we try to make, bottom line, when we try to make antibodies to fight viruses, it's tough because while we're still trying to make them, they're developing new antigens already. So once we get an antibody, it no longer recognizes the original antigen. So how do we fight them? This is one line. This is, we've all heard of AZT, right? So AZT, or this is actually AZC, this is cytosine up here. But what these drugs do is they're incorporated into DNA. Now, reverse transcriptases will bind this analog, right? This is an analog of a nucleotide. This is the ribose, and there's a phosphate here. Why isn't there phosphate on this guy here? Anybody know?
To prevent us.
It's in there. So when we treat cells, you know, what you want to do, if someone has AIDS, is give them this drug, AZT or ACT. It has to get into the cell. And drugs, phosphorylated molecules can't get through the membrane. So we give them just AZT or ADT, ADC, idioxycytidine, but not the T, not the triphosphate. It goes into cells. Once it's in, this 5' carbon of the ribose is triphosphorylated. So the cell phosphorylates it. So now it's really ADCT triphosphate or whatever. Anyway, once it's in the cell, reverse transcriptases can recognize that as one of their substrates, and they can put it into DNA. DNA polymerase doesn't. DNA polymerase won't bind this, won't fit in the substrate proper. So we can use these to treat viral infections, right? And it doesn't hurt the host cell because the regular DNA polymerases that replicate host DNA don't even bind to these guys. Only the retroviruses do. So this gets into the DNA, it's incorporated into DNA through its 5' phosphate. But then what happens is it has this linkage down here, it has this azeto group down here, and rather than a hydroxyl at the 3' position, the 3' carbon, there's no hydroxyl, there's this azeto group, so we can't add a new nucleotide to it, right? We can't take a new incoming nucleotide triphosphate and make a phosphoester bond. So it stops DNA synthesis. Works awesome. What's the problem? Well, these viruses mutate like crazy. So when you do this here, after enough viruses are there, that polymerase will mutate so it no longer binds to these, and this becomes ineffective. You generate a population of bacteria, don't bind to this drug, and it becomes useless again. See that? How do we get around that? Well, the drugs that we use for AIDS now, right, are cocktails. They have 3 or more inhibitors, and the chances of mutation occurring where all 3 inhibitors become inactive is really very slim. 3 or more even. Okay, so that's the cocktail deal. So that's reverse transcriptase. What happens to DNA? This is just the rest of the viral story. Once that virus RNA is reverse transcribed into double-stranded DNA, the double-stranded DNA becomes inserted into the genome. So now it's actually part of your host genome. It can sit there forever. A lot of people have hepatitis, they go through a treatment, they're cured, and they seem fine, and 30 years later they develop hepatomas, liver tumors. So it can sit there forever. Something happens along the way where this thing that's in there begins to be expressed. The gene nearby is turned on, it's translocated, so it moves into a different part of the chromosome in front of a promoter. Different things can happen, but once it's in front of a promoter, then this becomes actively transcribed into RNA. Some of that RNA serves as template for protein synthesis to make proteins, capsid proteins. These are the envelope proteins, those GP120 proteins, the reverse transcriptase, the enzyme itself. Some of them bind to a piece of tRNA to make a primer. assemble in the cell to form that virus inside the capsid, and then the capsid buds out of the cell. And as it forms a bud, it comes out, the host membrane comes around and becomes a little membrane in the cell. That's the total cycle for the RNA tumor viruses. So how do these things get into cells and out of cells? We talked a little bit about transposable elements. Remember we talked about wiring of lipid and the ACPC thing. So the did we do that here? I think we did. So the transposable elements can move around in DNA. And the reason they do is that the transposable elements, they have on their ends, they have these inverted repeats that allow them to move around. But also they code for genes. And one of the genes they code for is an enzyme that's called an integrase. Integrase, right? And those integrases are able to cut this out of the host DNA, and that cutting generates these inverted repeats. It's able to cut DNA in another location, and it cuts it in this, it makes this uneven cut, so you can see there's an end and there's an end. And then it inserts that in between, so this will get inserted in between. Integrase does that, then DNA polymerase fills in, and this thing is moved around. And the way we can find this, right, is because it cuts unevenly like this, right? So you have a TACG on this side and this guy on here. When the polymerase fills those in, we end up with direct repeats on the ends of these things, right? GTACG, DTACG. And people who are interested in transposons and how they move around find them in a genome simply by searching for these direct repeats to show where it's been inserted. And I told you before, these things move around. They can move around very freely in a genome and really take over large parts of it. Okay. I don't know if I have time, but I'm going to start. We talked about DNA damage. We talked about DNA 1, polymerase 1 in E. coli that repairs damage. We call it fragment damage. But there's other kinds of damages that occur to DNA all the time. This represents damage. All of these arrows can represent different kinds of damages, right? So these red arrows are all oxidative damage. Oxidation can open up these aromatic rings in the base. There's obviously ruins it. These blue arrows here are hydrolytic damage. So DNA that's in a cell can have hydrolysis occur a lot of times. If it occurs here, which you can see the thickness of this arrow is how often it occurs frequently here. But this hydrolysis of this here releases the base. And this is a purine, so if you release this, then the DNA at this site becomes what we call apurinic without the purity. Right? If you do it here, it becomes apyrimidinic. A very common one is the loss of this amino group or this amide on cytosine, right? The deamination of cytosine. You can see a pretty big arrow there. So these damages occur spontaneously in cells. This thing here, this depurination event, primarily in adenosine, more in adenosine, happens 1,000 times a day per cell. So the cells in your body are having these things are being depurinated 1,000 times. Every time, that's a chance for mutation. This cytosine, right, is being deaminated about 100 times a day per cell. What happens? What happens when this is deaminated? It becomes a what? If you remove this amine group and you put an oxygen there, it becomes uracil. So it's deaminated, it becomes a uracil. If that's left there, then when this strand replicates, rather than being a CG pair, replicating to a G, the uracil to an A, and then the next time around that A to a T, and you've changed a CG pair for an AT pair. 100 times a day per cell. 100 chances for every cell, most of the cells in your body, to develop a mutation. So they'll have to be corrected, and we have ways to correct them. This just shows you, this is a slide that shows you deamination. So that deaminization event leaves a uracil in there, and your body has enzymes that can detect that. We run some enzymes called glycosylases that remove the base, that cut the base off of a nucleotide base, a mononucleotide, right? So this is a uracil mononucleotide up there. And they cut the base off. How do they do it? Well, those glycosylases are very specific, and your cell has what's called uracil glycosylase. And what it does is it gets on your DNA and it walks along the DNA and it looks for mismatches. It looks for this uracil guanidine mismatch. How does it find those mismatches? How does it find them? How does it recognize them?
Hydrogen bond.
They're not hydrogen bonded, right? The GC-based pair is hydrogen bonded, but when it changed to uracil, it's no longer hydrogen bonded. That amino group is involved in hydrogen bonding. Now you put an oxygen, you're changing the position for a hydrogen bond. So here's what happens. This is this crystal structure. And what happens is that these glycosylases, uracil glycosylase, starts to walk around here and it tries to flip out the bases. It tries to flip them. If they have hydrogen bonding, it can't. If there's a uracil one that doesn't belong, it flips out and then it cuts it off. Right? And what happens when you do that, you end up with what we call depurinated. So here's a depurinated DNA. There's another group of enzymes, endonucleases. They're nucleases that can cut inside of the strand. One of them is called an AP endonuclease, which is an apurinic, or in this case, apyrimidinic. Right? You can do either one, nuclease. And there's a phosphodiesterase that cuts the phosphodiester bond on the other side. So it cuts the sugar phosphate backbone on both sides of where that base used to be, and it spits it out. Once it does, we have a template, right? And that template can serve then for DNA polymerase to put in the right nucleotide again, and then the ligase puts it together. Okay? This is called base excision repair, because what it does is it cuts the bad base off, and then cuts out, uses that endonuclease and the phosphodiesterase to cut the sugar phosphate backbone, right? You let that piece out, and then it just uses